Welcome to our series of COVID-19 webinars. We have created 4 so far to help understand more about this disease and the effects on patients. Please feel free to access all 4 webinars at a time which suits you. In Part 4, we will look at antibodies, immunity and vaccines as essential components of containing COVID-19 with everyone. Antibodies are what our own body produces against invading pathogens. Immunity is the body's ability to recognize something which is harmful and defend against this or kill this. The immune system includes various parts of the body, including the thymus, spleen, lymph nodes, special deposits of lymphoid tissue, such as those in the gastrointestinal tract and bone marrow, macrophages, lymphocytes, including the B cells and T cells, and antibodies. https://www.merriam-webster.com/dictionary/immunity-19th May 2020. There are 2 different types of immunity, active and passive. A vaccine can be a suspension of weakened, killed, or fragmented microorganisms or toxins, or of antibodies or lymphocytes that is administered primarily to prevent disease. So in order to understand what are antibodies and the role that they play, we need to examine pathogens. Pathogens are microorganisms and are generally referred to as causing harm to animals and plants. Common pathogens are bacteria, viruses, and fungi. We come into contact every day with microorganisms and they cause us no harm. Some are helpful, such as those in the gut which digest our food. So we know that COVID-19 is a disease. The disease is caused by a virus. The virus is called SARS-CoV-2. The virus is smaller than bacteria and cannot live on its own. It needs a host cell to complete their life cycle, and we are the current host for this global pandemic. This is a definite pathogen, as it causes harm to human beings, and we also know that the severity of its harm varies from person to person. You are probably aware of different pathogens, such as bacteria, viruses, fungi, and protozoa. These pathogens cause different human diseases as shown here. Let's take, for example, bacteria, which can cause tuberculosis, or tetanus, or a virus which can cause a common cold or the flu. Over the centuries, humans have encountered many pathogens, examined pathogens, and now understand how to recognize the symptoms of a disease caused by a pathogen and how to treat it effectively. The more data, studies, publications, clinical trials that we obtain about COVID-19, the better we will be able to comprehend and treat the virus. An antigen is a prominent part on the viral surface of a pathogen. This antigen is a molecule that can stimulate immune responses in our body and can bind with a specific antibody, while a pathogen is a harmful organism that causes disease in its host. Thus, this is the key difference between antigen and pathogen. Furthermore, a pathogen is an organism, but an antigen is not an organism. It is a molecule located in the cell wall of bacteria or the coating of other microorganisms. A pathogen is a microbe that makes us sick. In contrast, an antigen is part of a pathogen that is capable of stimulating an immune response in our body. Thus, this is the key difference between antigen and pathogen. Furthermore, antigens are proteins, peptides, polysaccharides, lipids, etc., while pathogens can be viruses, bacteria, fungi, protozoans, etc. The key difference between antigen and pathogen is that antigen is a foreign substance, toxin, or a molecule that can stimulate an immune response in order to produce antibodies against it, while pathogen is an organism, especially a microbe, that infects our body and causes diseases. Lymphocytes are part of our immune system and produce antibodies. By recognizing the antigen as being foreign, they produce antibodies that are specific to that antigen. This creates memory cells. After fighting infection, copies of the lymphocyte remain, so that if the pathogen returns, the body can immediately produce antibodies. Through our human evolution, we have learned from studying various pathogens that they themselves have learned to avoid immune responses. Take the influenza virus, for example. It constantly mutates its genes, thereby changing the antigens the immune system can recognize and allowing the survival of these newly mutated virus versions. The process, known as antigenic drift or antigenic variation, is also the reason why we have to vaccinate against the virus every year. Here we can see a lymphocyte on the left. It has antibodies attached to it due to the presence of a new or familiar pathogen with antigens protruding from it. The antibody is unique to that specific antigen, and each of our lymphocytes can only make one type of antibody. Now, this may take some time for our body to produce the correct antibody, and during this time, the pathogens can cause havoc, invading our cells and making us sick. We really need the antibodies produced as quickly as we can. Antibodies are Y-shaped and only attach themselves to the antigen. This then disrupts the pathogen, making it easier for other cells, called phagocytes, to find the now injured pathogen. Phagocytes are part of our immune system and will come to ingest and destroy invading pathogens. The phagocyte, may be a neutrophil or a monocyte, will surround the bacterial cell, enclosing it in a vacuole. Enzymes are secreted into the vacuole to destroy the bacterial cell. The process of ingesting the pathogen is called phagocytosis. If I have antibodies for SARS-CoV-2, can I be reinfected or pass infection to someone else? The immune response, including duration of immunity to SARS-CoV-2 infection, is not yet understood. Patients with MERS-CoV are unlikely to be reinfected shortly after they recover, but it is not yet known whether similar immune protection will be observed for patients with COVID-19. Now we can look at what is immunity. Immunization, process by which resistance to disease is acquired or induced in plants and animals. Immunization may occur naturally, as when a person is exposed unintentionally to a pathogen, any infectious agent, or it may be brought about artificially through a vaccine. In either case, immunization provides resistance or immunity to a particular pathogen by means of antibody proteins that are targeted to eliminate that pathogen from the body. As we have already seen, antigens are proteins that are found on the surface of the pathogen. Antigens are unique to that pathogen. Passive immunization imparts immediate but not long-lasting protection against a pathogen and may arise naturally, such as when a fetus receives antibodies from the mother across the placenta, or when a breastfeeding infant ingests antibodies in the mother's milk. Passive immunization against a particular pathogen, such as the hepatitis B virus, HBV, can also be conferred artificially. A person lacking immunity to HBV can receive a preparation called immune serum globulin that contains antibodies formed against the virus. These antibodies are obtained from serum taken from animal or human donors who previously were infected by or immunized against the virus. Active immunization stimulates the immune system to produce antibodies against a particular infectious agent. Active immunity can arise naturally, as when someone is exposed to a pathogen. For example, an individual who recovers from a first case of the measles is immune to further infection by the measles-causing virus because the virus stimulates the immune system to produce antibodies that specifically recognize and neutralize the pathogen the next time it is encountered. The term herd immunity refers to a reduction in the likelihood of someone catching a specific infectious disease because a significant proportion of the people in that person's community are immune to it. If you are less likely to come into contact with an infectious person, you're less likely to get infected yourself. The people in a community can be immune to a particular disease, especially through previous exposure or vaccination. Having a vaccine for COVID-19 is one of the ways to manage this disease. Vaccine, suspension of weakened, killed, or fragmented microorganisms or toxins or of antibodies or lymphocytes that is administered primarily to prevent disease. A vaccine can confer active immunity against a specific harmful agent by stimulating the immune system to attack the agent. Once stimulated by a vaccine, the antibody-producing cells, called B cells or B lymphocytes, remain sensitized and ready to respond to the agent should it ever gain entry to the body. Since the start of the pandemic, there has been an urgent need to accelerate the research and development of COVID-19 candidate vaccines. The World Health Organization has been supporting this effort. Currently, over 120 candidate vaccines have been mapped, and sites in 40 countries have expressed an interest to join the Vaccine Solidarity Trial. Here we can see 8 candidate vaccines in clinical evaluations at the time of the 11th of May. With a potential vaccine against coronavirus disease 2019, COVID-19, many months away, one proposal that some governments have suggested, including Chile, Germany, Italy, the UK, and the USA, is the use of immunity passports, i.e., digital or physical documents that certify an individual has been infected and is purportedly immune to SARS-CoV-2. Individuals in possession of an immunity passport could be exempt from physical restrictions and could return to work, school, and daily life. However, immunity passports pose considerable scientific, practical, equitable, and legal challenges. The World Health Organization issued the following statement. Some governments have suggested that the detection of antibodies to the SARS-CoV-2, the virus that causes COVID-19, could serve as the basis for an immunity passport or risk-free certificate that would enable individuals to travel or to return to work, assuming that they are protected against reinfection. There is currently no evidence that people who have recovered from COVID-19 and have antibodies are protected from a second infection. As of the 24th of April 2020, no study has evaluated whether the presence of antibodies to SARS-CoV-2 confers immunity to subsequent infection by this virus in humans. People who assume they are immune to a second infection because they've received a positive test result may ignore public health advice. Medela has recognized the challenges ahead for many countries, their populations, and healthcare systems. We want to reassure our customers that we are here to help them with the care of COVID-19 patients. We are preparing portable suction devices with virus filters and disposable canisters to cope with any second surges of COVID-19. As we have heard, we need our immune systems to gain immunity to COVID-19. Therefore, we need a vaccine to manage this process. All the ongoing trials and clinical research will be crucial to finding the right vaccine. At any moment's notice, clinicians, hospitals, and healthcare centers will have to quickly respond to future surges of COVID-19. Thank you for listening.